Human memories can remain vivid for over 70 years, yet the biological hardware storing them replaces itself constantly. To solve this paradox of longevity, we have to look past the fluid environment inside the neuron and examine the extracellular matrix. The extracellular matrix is a complex network of proteins and carbohydrates that surrounds, supports, and gives structure to cells and tissues in the body. Standard neurobiological consensus suggests that long-term memories are stored via intracellular molecular switches, like KMKI, inside postsynaptic spines. KMKI is a specialized enzyme inside neurons that facilitates the strengthening of synaptic connections during learning. This model faces a mathematical reality. These intracellular proteins have metabolic half-lives of mere days or weeks. Storing a memory for decades would require these proteins to be destroyed and recopied tens of thousands of times without ever introducing a structural error. True data longevity requires highly stable physical media. A computer hard drive does not rely on continuous, energy-dependent recopying of temporary electrical states. It writes the data into a rigid physical architecture. The human body possesses highly stable, long-lived proteins capable of acting as this permanent hard drive, but they are located completely outside the cell. Early anatomical observations of the central nervous system documented this rigid architecture. Santiago Ramon y Cajal and his contemporaries identified complex structures wrapping around neurons, though their potential role in memory was overlooked for over a century. The brain possesses a specialized sector of this matrix known as the perineuronal net, or PNN. The PNN is a dense, proteoglycan-rich extracellular structure that physically encases mature neurons and strictly limits the formation of new synapses. Nobel laureate Roger Tsien proposed that memories are stored as a precise pattern of physical holes, permanently carved into the surrounding perineuronal net. To solve the lifespan paradox of memory, we must move beyond the unstable recycling environment inside the cell and decode the rigid structural scaffolding outside of it. At the end of critical developmental periods, the perineuronal net hardens into a continuous restrictive barrier that locks neural wiring in place. This creates a physical challenge for neuroplasticity. For an adult brain to form a new memory or a synaptic connection, the neuron must physically break through this rigid matrix. The brain achieves this erosion using matrix metalloproteinases, specifically MMP9, which are rapidly secreted at synapses in response to intense neural activity. MMP9 is a specialized enzyme designed to sever the tough peptide bonds of extracellular matrix proteins. This localized proteolytic digestion creates perfectly sized gaps in the matrix, allowing specific synaptic spines to enlarge and connect. The PNN functions as a fantastically convoluted 3D punched card. The precise three-dimensional coordinate and size of every hole permanently records the location and strength of a synapse. Administering targeted MMP9 inhibitors prevents the formation of late-phase long-term potentiation and stops the acquisition of specific fear memories. Long-term potentiation is a persistent strengthening of synapses based on recent patterns of intense electrical activity. Proving this hypothesis requires demonstrating that the proteins forming the perineuronal net outlive the proteins inside the synapse itself. If MMP9 carves architectural changes into a stable matrix, memory is an act of structural sculpting, moving the engram from a temporary electrical event to a permanent physical fossil. Researchers face a logistical challenge in determining the metabolic age of a microscopic protein structure buried deep inside a living brain. One methodology involves establishing a baseline of old proteins during early development using 15N isotope diets in mice. 15N is a stable, heavy isotope of nitrogen that researchers use to trace the exact age and synthesis rate of proteins in an organism. Researchers use mud pit mass spectrometry to separate and compare the 15N retention of perineuronal net proteins versus synaptic proteins months later. Mud pit is a multidimensional liquid chromatography technique that separates and identifies massive numbers of complex peptides simultaneously. While effective in animal models, isotope feeding cannot be used to birth date proteins in living human subjects. A historical anomaly provides a global biological clock. the massive spike in atmospheric carbon-14 caused by nuclear weapons testing in the mid-20th century. Carbon-14 is a radioactive isotope of carbon that decays at a known rate, allowing scientists to determine the exact age of organic materials. This carbon-14 pulse was incorporated into the long-lived proteins of the global population. By applying accelerator mass spectrometry to human cadavers, scientists can measure this isotope to prove that PNN components last for decades without replacement. Confirming the longevity of the matrix is only half the proof. Researchers must also observe the active creation of these memory holes in real time by tagging the exact neurons firing during a learning event.
To do this, researchers design an optogenetic snapshot reporter. Optogenetics is a biological technique that uses targeted light to control the activity of genetically modified cells in living tissue. A snapshot reporter is a highly specific engineered genetic tool designed to capture and permanently record a single biological event precisely when it happens. This coincidence requirement, needing both a biological trigger and an external light trigger, allows researchers to permanently tag and exert optogenetic control over only the specific neural ensemble active at the exact moment a fear memory is formed. By merging the radiocarbon birth dating of the physical matrix with optogenetic snapshot reporters mapping the neural activity, researchers can map the physical architecture of a specific memory. Expanding the frame. The brain's architecture does not end at the neck. Structural memory is not an exclusively neurological phenomenon. Fascia acts as the peripheral equivalent of the perineuronal net. Fascia is the continuous connective tissue matrix that wholly envelops and structurally integrates all muscles, organs, and nerves in the body. Fascia stores physical information through tensional memory. Tensional memory is the biological process where mechanical forces dictate exactly where new collagen is deposited along lines of strain. Mechanical forces from chronic posture or physical trauma generate a permanent architectural record. This creates a physical, medium-term memory of the biomechanical loads the organism has endured. Fibroblasts are the primary cells embedded within connective tissue that actively synthesize the extracellular matrix and monitor its mechanical tension. Fibroblasts sense the rigidity of their extracellular microenvironment and exhibit epigenetic memory. Epigenetic memory refers to heritable changes in gene expression and cell behavior that occur without altering the underlying DNA sequence. In fascia, external mechanical signals alter chromatin architecture inside the cell, creating permanent changes in how the tissue responds to inflammation. This systemic response relies on the biomechanical concept of tensegrity. Tensegrity is a structural principle where an entire system is stabilized by a continuous web of tension cables and discontinuous compression struts. Because the fascial web operates as a tensegrity network, localized physical trauma creates a ripple effect. Pulling on the matrix in one location automatically transmits structural memory and tension across the entire organism. This communication is incredibly fast due to the piezoelectric properties of crystalline collagen fibers. Piezoelectricity is the electric charge that accumulates in certain solid materials in response to applied mechanical stress. This allows the fascial web to generate rapid, body-wide vibrational communication signals under physical tension. The fascial web acts as a continuous biomechanical hard drive. Peripheral trauma is written into the body's collagen architecture, just as neural memories are etched into the perineuronal net. These two scales of extracellular matrix, the specialized neural nets and the systemic fascial web, provide an informational network extending across the entire organism. If pathological memories, including traumatic psychological associations and chronic physical pain, are structurally locked in this rigid matrix, how do we overwrite them? Manual interventions can trigger a biological mechanism known as gel-sol transformation. Gel-sol transformation is a physical state change in the fascial ground substance from a dense, rigid gel to a fluid solution, driven entirely by targeted mechanical pressure and temperature changes. Manual therapy and physical pressure applied to dysfunctional tissues physically depolymerize microtubules within the matrix. Microtubules are rigid, tubular protein polymers that form the structural cytoskeleton of a cell and dictate its physical shape. This physical shift from a dense gel to a fluid sol state erases the physical memory traces stored in the tissue's architecture. It flushes out trapped inflammatory cytokines. Cytokines are small proteins released by cells that directly control the growth and activity of the immune system and drive local inflammation. This localized peripheral release directly impacts the central nervous system. The fluid transformation decreases the chemical irritation of autonomic nerve endings embedded in the tissue, sending a massive reset signal straight back to the spinal cord and the brain. This structural erasure in the periphery can provoke sudden somato-emotional phenomena. When the physical matrix releases its tension, it can trigger the spontaneous recall and sudden release of deeply held traumatic memories. Memory exists as a permanent interference pattern etched into the structural architecture of the entire organism. To understand, heal, or alter the memories that define us, science and medicine must move beyond the fluid inside the cells and learn to read and rewrite the physical scaffolding of the human body.